We are live now, and good morning, everyone. All right, so a couple of things to think about. We are getting close to the first exam, and exactly when it's going to be, it'll either be Friday or Monday, and I can't tell you which yet, but probably by the end of the day I'll know. I'm thinking Monday. The exam, I think the format's going to be not in person. My plan is to drop it at 8 a.m. on Monday morning, and then it'll be due at 10 a.m. Monday morning. So plan accordingly. It's only designed to be an hour long, but you got 2 hours. And if you have to look everything up on Google, well, that might take a little bit longer time. I don't know. I'll let you figure that out. But it's going to be open notes, open everything. You just can't work with other people. And yeah. Oh, then there's also, we're going to be getting into protein translation today. And another thing that will drop this week will be a little homework assignment that we'll talk a little bit more as we get into it. Okay. Dramatic pause for coffee. Thank you. And so we finished up talking about the mechanisms or the beginnings of translation. And I think the last bit we were talking about was the coding potential for nucleic acids. The fact that we know we have 20 amino acids, but 4 nucleotides. So the simplest approach would be 1 nucleotide equals 1 amino acid. That doesn't give you sufficient coding or information depth. The number of bits doesn't work. Then if you look at 2 nucleotides is equivalent to 1 amino acid, that's going to be 4 nucleotides, 2 each. The combinations are 16. Still not enough. If you go with 3 nucleotides equals 1 amino acid, that's going to be 4 cubed, and that's 64 possible combinations. Bing, we're there. Not only do we have enough for 20 amino acids, you could add a few extra amino acids if you wanted to, but we also have now some redundancy in that more than one nucleic acid, or an amino acid may be coded for by more than one 3 nucleotide pair. We call it codon. We're also going to have some codons that we use as stop codons. And so the ability now to think about how an amino acid polymer can be translated from the nucleotide sequence of 4 bases becomes reality. And a couple of things that we know then is that we're going to be looking at 3 nucleotides codes for 1 amino acid. So right off the bat, a couple of things that come to mind is that the DNA or RNA strand that codes for a gene is going to be a minimum 3 times the size of the amino acid polymer that it codes for. As it just turns out, the average amino acid molecular weight is about the same average molecular weight of a nucleotide. And so we know then that the RNA is going to be 3 times as big as the protein that it codes for at a minimum. So now we know this. The next step is to identify what is known as, or we can think of as the adapter molecule. I just made that term up. That is the tRNA. Transfer RNA. tRNAs are about 130 or so, 150 nucleotides, and they've got a structure that looks a little bit like this. So the 5' phosphate is over here, and it forms a kind of a cloverleaf structure in which we have intermolecular base pairing. And in fact, some of it is non-Watson and Crick. There's a couple of weird nucleotide modifications. Not that we need to worry about that. But it forms this sort of cruciform shape that then is able to actually kind of fold around and sort of hug itself into this three-dimensional shape of a tRNA. And at the 3' hydroxyl is where we've got the amino acid. And it's just something that, in terms of biochemistry and thinking about these things, it's easy to remember if you can think about the chemistry a little bit, in that we will always want to have a free amino terminus. That's the reactive end of the amino acid in terms of synthesizing it. So because of that, and if you remember, the amino acid looks like this, where we have the amine group and 2 hydrogens or 3, depending on the pH, not important. The central carbon and the carboxylic acid. And the R group here, well, that OH is what we're going to bind to. So the amino acid is linked covalently to the 3' end of this very long, 100 nucleotide RNA strand that folds up. And this is the link between the nucleic acid world and the amino acid world. And of course, hopefully from genetics or on the street, you know that here we're going to have 3 nucleotides specifically that are known as the anticodon, and they base pair with the messenger RNA. So the mRNA is going to be then defined in these 3 nucleotide chunks that we call the codon. So that's just the term we use for the 3 nucleotides that code for an amino acid. And once this started to get figured out back in the 50s, it then just becomes a matter of sort of slogging through and figuring out if you feed in to an in vitro translation system a poly-U track, just U. And you can then look to see that you generate a polyphenylalanine amino acid polymer. So we know that phenylalanine is coded for by a U. And people began biosynthesizing different RNA sequences of different repeats and things, and began to look to see what kind of amino acids are being generated. And from this, we generate our codon table. This is, there are a number of different ways of looking at it. You can have just sort of an XY chart and things, and there's a lot of codon tables around. There's one in the book. And what this does is gives you the lookup chart. So if your RNA has, this specific nucleotide sequence, you can decode it. Now, once we get into this kind of biz, a couple of things also begin to fall out that can make things quite confusing. And that has to do with the fact that because we're reading it 3 nucleotides at a time, we have what is known as the reading frame. So let's just look at this for a moment. If we have an arbitrary sequence, AAU, GGC, UUU, AGU, just as a sequence, and I'm not hip enough to have all my codons memorized. God forbid, if you're that big a nerd, mercy on your soul. I look it up, or I actually use a computer to translate it for me. But regardless, what we have then is a string of nucleotides. And if we begin sort of parsing through this, we can see some interesting issues start to arise in that we can break this into 3 nucleotides like so. I mean, I kind of always write my nucleotide strands in groups of 3 just because. So we have AAU, GGC, UUU, and AGU. Okay, so that would be a codon set. We can decode it, figure out what it is, good. But we can also step one over, because who's to say I'm the first nucleotide of the amino acid sequence? This is where we start. I mean, actually we know who's to say, but we'll get to that in just a moment. And we can have the sequence read in a second reading frame. And that would be starting over here with that A. And now we're going to have the triplets AUG, GCU, UUA, and GU. So that would be what's called the second reading frame. Then we can have another reading frame where we start out like this with A. This is going to be the first base, and we have UGG, CUU, see you later, UAG, and U. And, all right, if you just to make sure you want to check, we can have another reading frame where we start here, and that would be GGC, UUU, AGU. Hey, wait, that's the same as that one. Okay. All you did was just lose one codon. What that means is, we throw that guy out, we have 3 possible, what we would refer to as forward reading frames. And were we to use a codon lookup chart, we could figure out what the amino acids are going to be. And what I'll tell you is, if you are looking at the amino acid sequence in the next reading frame, they will be completely different. In the third reading frame, again, completely different. So the amino acid sequence that you generate is critically dependent on the reading frame. If you're in the wrong reading frame, you're not making the correct protein, and generally you're making garbage. So identifying the reading frame is a really critical point. From a computational perspective, if you go to my favorite program for doing reading frame analysis is something called EXPASY. I'm not sure if it's xpasy.com, but regardless, xpasy is one of the standard sequence translators that I like to use, and there's a ton of them on the web. I'm sure there's an app on your phone. And you plug in your sequence, and it goes boom, and it spits out all 3 reading frames. And then it's up to the user to figure out, all right, which one makes the best sense. And sometimes you can't tell. Often, what will happen is, and we'll see as we get into this, when you're in the wrong reading frame, there'll be stop codons, or codons that don't have an amino acid coupled to them. That's referred to as a stop codon or a nonsense codon. And you go, okay, that's probably not it. Or there may be a sequencing error. You have to kind of just sit there and start thinking about it. But this is a great place to have mutations. If you have a single nucleotide deletion, that will throw you off reading frame. 2 nucleotide deletion throws you off reading frame. A 3 nucleotide deletion doesn't throw you off reading frame, it just deletes one amino acid. And so there are all kinds of mutations that occur from reading frame shifts. And a reading frame shift, that's a bad mutation because you pretty much can't recover from that. All right, so the other aspect of computational genetics is when you're given a sequence, unless you're told, all right, the promoter is over here, and this is the top strand, in which case then you know to translate it. You also can have the possibility of the gene is going that way. And this is referred to as the reverse reading frames. So it's just a relative term, but there are always going to be 3 forwards and 3 reverses. So for a given DNA sequence, unless you're told explicitly, there are the possibility of 6 reading frames. 3 forward, 3 reverse. And this is what the homework's going to be, and I'll just give you a quick little hint or a tool. When you're given DNA sequence, go ahead and just start up again. the DNA sequence, of course, you don't have the uracil, you have thymine. And in terms of coding, T, U, whatever, I mean, it's not like it's a big deal. So we have G-A-A-T-T, G-A-A-T-C, A-G-C-A-A-T. This is a sequence. So that is going to be the sequence we'd use for the 3 forward reading frames. And so we just go through and identify the 3 possible reading frames. So GAA, TCA, GCA, AT. And then reading frame 2 would be AAT, CAG, CAA. And reading frame 3 would be ATC, AGC, AAT. Okay, so you go through and you figure out your 3 forward reading frames. To find the 3 reverse reading frames, this is the steps you take. First, you complement the sequence. Complement the sequence. You're a beautiful sequence. You're a very lovely sequence. I really like you a lot. Then you take the appropriate base pairing. That was a joke. That's what it is to complement the sequence. Sorry. You can tip your waiter later. So to complement the sequence, you're going to base pair it. So it's going to be CTT, AG, TC, GTTA. So that's the complement. That's the bottom strand. In order to actually make this thing sing, though, the second thing you do is write it backwards. So use a mirror, whatever. It's not going to be A, T, T, G, C, T, G, A, T, T, C. That's the 5 prime. That's the 3 prime. And when I'm in the middle of doing DNA sequence analysis and figuring out primers for PCR, which we'll talk about later, I fill the whiteboard up with sequence just like this and end up standing on my head and kind of doing all kinds of things. And so now once you've got the reverse complement, and I'll also say I use a program called SnapGene, and you can put the sequence into the box, and then there's a little check box that says reverse complement. If you hit that, it just does it for you. But doing it by hand is useful. And so now we go through and do the same kind of reading frames. AAT, GCT, GAT, TC, TTG, CTG, ATTC, and TGC, TGA, TTC. Okay. That's how you then identify all 6 reading frames. And you then go through and translate them. So this is a little point in the homework. You're going to be given a 20 or so nucleotide sequence. You're going to generate all 3 forward reading frames, reverse complement it, generate all 3 reverse reading frames, and then decode each one. It takes a little while. You'll see in a moment we have these things that are called stop codons. These are codons that don't have a corresponding tRNA. And anything that has a stop codon, you don't have to mess with. The ones that are clean open reading frames that you have good, for the codons you have good amino acid sequence, we're going to actually run them through a database and see what they correspond to, see if you can identify the appropriate gene. We'll talk about the homework later. All right, so that's how we set this thing up. And the tRNAs then are critical in that they are base pairing with the 3 nucleotides, and they are bringing in the individual amino acids. Now, let's see a couple of points here. There is an enzyme system that is known as the tRNA amino acyl synthase. And these are pretty big protein complexes. They are responsible for identifying the tRNA, looking at the anticodon, and putting the appropriate amino acid on them. And these guys are pretty darn accurate. This process is reasonably slow, and it's got a very high degree of accuracy. Because, as we're about to see here, the failure rate or the error rate of this system is really pretty darn low. And thankfully, it's important because of protein structure that we have the correct amino acid. You could throw a monkey wrench into things if you end up having an inappropriate amino acid polymer. It can be folded up incorrectly. And so the primary spot where error checking is occurring is here. So we take great pains to very carefully look at that tRNA, eyeball it carefully, double check the anticodon, stick the appropriate amino acid on it. All right, so there are what are referred to as iso-accepting systems, in that there are amino acids, as we start looking at the chart, and we may throw it up on the screen here in a little bit, that an amino acid that has multiple codons that code for it. One of the standard things when we start looking at this is very often, so just as an example, CGU, and I'm not sure what this codon codes for, but I'm just throwing this one as an example. Let's just say that it codes for the amino acid tryptophan. And again, I probably should be looking at the book rather than pulling this out of my ear, but whatever. Very often, it's the first two nucleotides that are critical for the identification. And this third one could be a U, it could be a G, it could be an A, it could be a C. And this is an example. I will find another one I can say, yeah, this is it later, but not worry about it. Meaning that we've got the first two codons are critical. The first two nucleotides are critical. The third nucleotide actually is not critical. It can be anything, kind of a wild card. This is what's referred to as the wobble mechanism, in that the first two nucleotides of a codon are really critical most of the time. There are a few that are single nucleotide or single codon systems that, just as an example, the codon AUG, that codes for methionine, and nothing else does. So there's a fair amount, because we've got 64 combinations, we got a fair amount of redundancy as well. Additionally, as we'll see here in a bit, there are codons, and oh geez, some of the stuff is like, I should look this up, or I should have it in my head better, 3 or 4, not critical, what are known as stop codons. And really what these are is codons are nucleotide sequences that have no corresponding tRNA. And this is kind of important from an experimental perspective, in that you can modify a tRNA to hybridize to this stop codon, and then it's no longer a stop codon. But this allows us to stop translation, to halt translation. All right, so now that we've got a little bit of this stuff set up, and just as a little thought, are we deeply confused, or is everybody hip? Good. The next critical step is putting this all together. And let's think about the fact that because there are 3 open reading frames, and I mean, because the business about the reverse open reading frame, you settle that with making the RNA strand. That is, whichever direction the promoter is going. So with an RNA, a given RNA, there's 3 open reading frames. How do we decide which is the correct open reading frame? And that's actually a very important point. So when we start thinking about the production of an RNA, of a protein from an RNA, start thinking about translation. We've got these terms that we use that you'll see being, potentially familiar, and that we have initiation, elongation and termination all across the nation. So, in this case, we use these exact same terms talking about the production of an RNA. We don't have that much to say about elongation or termination. We got stuff to say about the translation system. A lot of alliteration in this system of translation and initiation and elongation. Somebody stop me. All right, so again, we've got some good comparisons between prokaryotes and eukaryotes. And the last player that we need to introduce in this lineup is this very large, maybe even considered an organelle, but what's known as a ribonucleoprotein, meaning that it is an RNA and it's covered with lots of proteins. And that is the organelle known as the ribosome. The ribosome is, as the name implies, a ribonucleoprotein. It has an RNA, and then it's got a bunch of proteins associated with it. And feel free to memorize the name and size of every protein associated with the RNA. No, don't. Why even bother? Ribosomes are made up of two parts. There's what's known as the small subunit and the large subunit. And they're given these terms of like 70S and 60S. These are now rather quaint ways of measuring sizes of really big molecules and really big ribonuclear proteins using centrifugation, density gradient centrifugation is referred to as a Svedberg unit. And when you add them up, they don't always add up right, which, it's like, really, do you need to memorize this? No. Just if you see the term 70S, that's what it means. So the large subunit and the small subunit. In our various diagrams, we typically draw the small subunit as something that looks a little bit like the bottom of a hamburger patty or of a hamburger bun, and the large subunit is drawn either as a turkey or chicken, or I tend to think of it as the top of the large hamburger bun. So that's my diagram. There you go. And then the RNA is going to fit in between. All right. So this is the ribosome complex. Now, when we begin translation, we have to initiate translation. And initiation makes use of what's known as the initiation complex. It's this. We're going to have the initiation complex, blah, 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 is the small subunit, so the bottom of our hamburger bun, and it's got attached to it a tRNA that I draw sort of a cartoon like this. This is the initiating tRNA, and pretty much most, if not all the time, not all the time, but most of the time, the first amino acid is going to be coded for the codon AUG. The first amino acid is a methionine. And so the initiation complex takes advantage of this, and it's got a methionine, tRNA, attached to it. There is a small initiating factor, an initiating factor, that is blocking the large subunit from binding. So this is keeping the large subunit from latching on. And this is our initiation complex. And what this is doing is floating around and looking for an RNA to begin translating. So we have the system sort of loaded up with the first tRNA that we're going to need, and it's floating around. Now, this is where we have a key difference between prokaryotes and eukaryotes, because up to this point, really everything's the same. The codon chart is the same, which is pretty kind of amazingly weird, but the codon chart is the same. That is, bacteria and animals speak the same genetic language. The ribosomes, while there's some structural differences and size differences, there's a large subunit and a small subunit. The small subunit is the initiating complex. But in the prokaryotic model, we've got a sequence in the RNA. So here's our RNA sequence that is known as the ribosome binding site, or the ribosome binding sequence. And it turns out, this is sort of a nerd point, that the small subunit has a complementary sequence to this sequence. This sequence is also given the name of, I guess, the two dudes that discovered it. It is known as the Shine-Dalgarno sequence. Dalgarno didn't send me a Christmas card last year, so I said to hell with him. I don't call it the Shine-Dalgarno sequence. I call it the ribosome binding site. Whatever. No. Shine and Dalgarno are long dead. They don't know me. They don't owe me a Christmas card. There you go. But the ribosome binding site. And this is, as the name implies, the site where the small subunit binds. The ribosome binding site is placed in such a way that right next to it, a few nucleotides down, is the sequence AUG. This is the initiating methionine. And this is how we establish what is the correct open reading frame. Now, just as a little point, you might have AUGs scattered around just by chance, but if they're not in the appropriate context right next to the ribosome binding site, they're not the AUG that is considered it. So, the small subunit binds here, sets up shop. We're going to erase for a second and get a little bit better diagram. And it begins translation. So we can think of it now like this. We've got the 5', here's the AUG, the small subunit and its tRNA binds here with the methionine. The initiation factor, once this thing kind of links up, it diffuses away. And we now have the large subunit come landing. Boom. All right. The large subunit, when it binds, it creates the complete translation machinery now. And the large subunit has what I think of it as a little bit like 3 alcoves. If you recall, I had mentioned that one of the prerequisites for this class was to be familiar with Star Trek episodes. And so hopefully you've been doing your homework and you know of the Borg. If they have to go into their regeneration alcoves, same thing. Not really, but regardless. It looks like this. We got the RNA here. We got the small subunit here. The big subunit, large subunit here. So we got the top of the hamburger bun, the bottom of the hamburger bun, and then this here is the sliding meat. I don't know, it's a very long, very skinny hamburger patty. And we have 3 sites here. The first site is known as the E site. The second site is known as the P site, and not inconsequentially, the tRNA is sitting here with methionine. And the next site is known as the A site. So we have the EPA, the Environmental Protection Agency. What these really mean, though, is we have the exit site for the D-aminoacylated tRNA to move out, we've got the peptidyl synthase site, which is the P site, and the acceptor site. Okay, so a lot of things happening here. When we set up with the initiation factors has diffused away, we have the initiating methionine in place. We've got the system set up on top of the initiating methionine, the AUG. The next codon is hanging right here. And tRNAs are frantically slamming around in here. And when a tRNA can hybridize, it sticks. So one of the points is that in terms of error checking, this is happening pretty fast. And so there's not really a whole lot of chance for error. And so it's another one of these spots that's a fairly accurate place. Because if you don't have binding affinity, man, you get thrown out. So the next tRNA slams in here and hybridizes. And so it's carrying the amino acid number 2. Now, the enzyme that couples these two amino acids together is known as peptidyl synthase. It requires an ATP molecule, so we're going to use and dephosphorylate an ATP to generate the energy to make that. The peptidyl synthase is not a separate floating around enzyme. It's actually integral into the large subunit. So the large subunit has the ability to catalyze the production of the peptide bond. And we're not going to go into too much of a high mag or belabor the point, but remember, the way the amino acid is hooked on here, the carboxy terminus is attached to the tRNA. The amino terminus is here, the amino end. So the amino end is floating out here. We're going to have a joining. Bing! We make the peptide bond. And what we're going to do is essentially pop the amino acid off of the amino acid in the P site and couple it to the amino acid that is attached to the tRNA in the A site. And the next step then is the elongation step. So one way to think about this is we've got these two tRNAs sitting here, kind of lined up everything. And the next step is ka-chunk. The large subunit just moves over. So you can think of it either direction, but it's not like this is being pulled through. The large subunit goes kaboom, and it moves over, and it essentially is keeping the alignment the tRNAs have established and just going kaboom and chunking over one. And so if we were to do this with a bit of blackboard animation, it's going to look like this. So that is now cut loose. So we got the D-aminoacylated site. Bam. Here is E, here is P, and here's the A site, empty and waiting for something to happen. The small subunit moves right along right behind it, and we have moved forward 3 nucleotides at a time. It's a little bit like having a jig that you're using to kind of register if you're making a joint or something. There's all kinds of analogies, whatever you like. In that the presence of these tRNAs is making a structure that the large subunit can move around. So it just moves over to the one next site. If you don't have a protein associated with you, if you're stuck in the E site, all of a sudden your binding affinity is not very good, and you are cut loose. So here is the D-aminoacylated tRNA floats free, just like that. And we now have the tRNA for amino acid number 3 comes in, and the process just keeps on going. It takes a little while, but you can chunk through an RNA in a couple of minutes. And so a really large protein that might have, say 1,000 amino acids, it'll be around 3,000 nucleotides, and it'll probably take 3 or 4 minutes to actually be synthesized. And as this is happening, and this guy is chunking down the road, as soon as the ribosome binding site is cleared, another set can come on and start its road. So you have, with one RNA, very quickly it gets loaded with ribosomes. Boom, 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 boom. And the guy at the end, he's just about done. The guy at the beginning is just starting the road, and this system works. Now, the last step in this is termination. So termination is a cool trick. Termination happens when we run across what is known as a stop codon. Stop codons, and God, this is bad because there's these little factoids that, because I don't have my book in front of me, that I'm like, is it 3 or 4? It's not important. 3, 4. There is a set of stop codons. And a stop codon ain't anything special. It is simply a nucleotide combination that has no corresponding tRNA. And so when the ribosome hits at the A site one of these stop codons, it just hangs, it stalls, because there's no tRNA to slam in there. When the stalling goes on long enough, it's like, okay, we're going to give you 10 seconds. If nobody comes in there, that must be a stop codon. There is a termination factor, another protein, that is kind of waiting around, always looking to get in. The termination factor, this is a protein. It actually has a shape very similar to the tRNA A site structure. It just slams in there, and it is essentially a trigger and probably a hydrolase of some kind that pops the amino acid polymer from the tRNA and the P site and says, go free, you are now a fully translated protein, it diffuses away. So a key point is a stop codon is a stop codon because there's no tRNA. In fact, what you can do is have, at least in bacteria, in eukaryotes, at least in complicated eukaryotes, you don't have this. You can have something called a stop codon suppressor mutation. And all this is a mutation in a tRNA. It's a tRNA that recognizes the stop codon. And so the stop codon no longer works. And because it's a tRNA that lands in there, and it can be a variety of different amino acids, whatever. The mutation labeling will show that. And that will allow for read-through. We don't have that. Generally, when you get to the end of the reading frame, you'll have a stop codon, and then it'll be followed by another stop codon, and then it'll be followed by another stop codon. And so when you start looking at translation, open reading frames are pretty special. And when you see a sweet open reading frame, it is just like, straight amino acids, just like, yeah, this is it. And then when you're not in the reading frame, it's like stop, stop, stop. There's all kinds of stops. So this is prokaryotic translation. And it is darn similar to eukaryotic translation, but there are a couple of key differences that we will pick up on Wednesday. Yay. Questions, thoughts? Keep them to yourself. Post them in the comment section.